And so without further ado, let's move on to our second talk. That's going to be given by Greg Forrest. Greg, go ahead.
Okay, I believe Bruce is going to share my screen. I have a very weak Wi-Fi, so if I phase in and out, just let me know. So I'm Greg Forrest from UNC Chapel Hill. The two folks that I'm mostly driving this work through, 3 grants that are listed at the bottom here, are Ronit Freeman, who's been here for 3 years. She's a synthetic chemist, a so-called DNA nanotechnologist, who we brought in to study mucus pathology, and Sam Lai, who's been here about 10 years, came from Hopkins, who did a lot of work with Richard Cohn and Justin Haynes on studying HIV and other viral pathogens. So I'm going to talk about work that's, I'll mention later, but we've been working with the Multiscale Lung Institute for over 20 years now, and we're trying to bring a lot of the things that we've learned to bear on mechanistic modeling of the outcomes that were observed in our lung institute from SARS-CoV-2. Next slide, please. Bruce, can you go to the next slide?
Seems to be taking a few minutes to advance, I think.
Okay. I have such a weak Wi-Fi that I couldn't show the slides myself, everyone.
There we go.
Okay, the team is, I brought back folks that had been at UNC. Alex Chen, from the first time I met Sam Lai, and we were studying antibody virus mucus interactions. Alex is going on, and now he's at Cal State in Dominguez Hills. Tim Wessler, who graduated with me, also worked with Alex and a bunch of other folks on antibody virus mucus interactions. He's been at Michigan for the last 3 years. My student, Kate Daftari, who's a 3rd-year applied math student, and then a student of Ronit Freeman. The folks that give us all the intel, Rick Boucher I've worked with. We started something called the Virtual Lung Project 22 years ago, all across UNC, chemists, physicists, applied mathematicians, and a bunch of medical folks. And then another person in the Multiscale Lung Institute is Ray Pickles, who's an expert on respiratory viruses and lung infections. Next slide, please. Okay, our model, it's very complementary to what Morgan talked about. We've worked a lot on what happens when your mucus is pathological, or when you are exposed to a virus and have antibodies, and now we're dealing with when you don't have any immune protection. So that's what our baseline is. We use detailed physiology of the human lung, from the nose all the way to the alveolar space, what liquids line those surfaces where the viruses will land when you inhale them, whether or not those particular generations are transporting by mucociliary clearance, what percentage of cells in every branch are infectable, and then the kinetics of SARS-CoV-2. We know its diffusivity. We know if it's in the layers that are being advected by mucociliary transport, how that's affecting their mobility. We know the probability of infection per encounter. That's like a proxy for ACE2 receptors, if you like. And then we know once a cell's infected by SARS-CoV-2, how many infectious virions does it replicate every day, and for how many days? 3 to 5, essentially, and then it bursts. And so the first thing we took our model to task on are 2 clinical observations that you probably all know well, namely how easy it is to get an upper respiratory nasal infection very rapidly after a minor exposure, and then how would either a high-titer, highly infectious nasal infection propagate to the deep lung. Next slide, please. So, by the way, I will, we don't have any of this work submitted yet. So I will get that sent to the archive and upload it in the IMAG website whenever we do. This lecture is making me accelerate that, hopefully. That we embed in our model. I mentioned briefly the respiratory tract physiology and geometry. So we know what the thicknesses of the liquids are throughout the lung. We know if they're transporting and how fast they're transporting, how things scale as you go from the upper respiratory tract down to the alveolar space. Then we have measurements of the kinetics of SARS-CoV-2, measure its diffusivity in those liquids, infectability of cells, the latency time. Once a cell's infected, it takes about 12 hours before it starts replicating. The replication rate, which turns out to be about 2,000 infectious virions per day, many, many more that are non-infectious, and that lasts for 3 to 5 days. And then we build in the exposures. Inhaled exposures from the outside, and then secondary exposures, which I'll talk about, which is where you basically infect yourself from a nasal infection. So many of these parameters vary dramatically across the population. So we establish bounds on these parameters in the tables for all 3 of these. And at the end, we can talk more about that, risk factors for subpopulations. And I will again make this data, it's very hard to get all this data, and we will make it available on the IMAG MSM website and then as publicly available as possible. Next slide, please, Bruce. So the first thing is, why with a minimal exposure would you get a nasal infection? So more than half of the nasal cells are ciliated and they are infectable. They're the infectable cells in the nasal tract. Furthermore, they have high density of ACE2 receptors and other properties which I'm less familiar with that make the likelihood of infection per encounter with an infectious virion very high, like 30%. Once infected, the cells are latent for about 12 hours. And then they replicate on the order of, as I said earlier, about 2,000 infectious virions a day for 3 to 5 days, and then they explode. Those virions, it's way more efficient for the viruses that the cells shed the infectious virions back into the airway surface liquid. Way more dominant in spread of infection than cell-to-cell contacts. And then we know what the mobility of these virions are. We measure that. Those conditions lead to, with super high probability, a high titer, 10 to the 6th infectious virions per mil in less than 2 days. Next slide, please. So here's the consequences of the model for this. The leftmost column is we're assuming that there's a very mild advection in the nasal passage. I have it listed up top, 0.06 millimeters per minute. That will bias the transport by the viruses. This is infection, and the color code is on how long the cells have been infected. The advection will bias the transport of the infection along the direction of the advection. And using all the data I showed you before, by the way, these dimensions are in cells, which have a medium diameter of about 4 microns. So if you wanted that to be dimensions. And then on the right, we show how within 2 days, the viral load is up to 10 to the 6 infectious virions from one infectious seed infecting one cell, and then it propagates. So one of the messages here is that how much you inhale in a different, some location is dominated very early by the replication of infected cells, of infectious virions by infected cells. Next slide, please, Bruce. If there was no drainage, if it was stationary, and it'll be slightly less. So advection actually accelerates the spread of the infection, but by a minor factor of about 5. Okay, next slide, Bruce. The second result is an important result, which is that when you get any type of an infectious seed in the respiratory tract, wherever there is advection of that mucus layer, as the last slide or 2 previous slides showed, the propagation of that infection is up, not down toward the alveolar space. And that has important implications. So we, in the figures below, you start out with a healthy cell. There's a seed somewhere in the respiratory tract. In this case, it was the very weak advection in generation 9. And you can see the black are the cells that have been infected and have gone through the latency and started shedding. The red are the infected cells that have not started shedding yet. And you can see it very weakly propagates down because of advection dominating diffusion. And then within a month, that's how far the infectious infection front has propagated. So depending on what generation is, how long that particular branch is, you can calculate how many branches would have been infected. Next slide. So the implications of this result above are profound. It says that if you're going to have a significant, infection in the alveolar space, such that you would have alveolar pneumonia, the infectious seeds have to be deposited directly in the deep lung. They are not making it there by upper respiratory or lower respiratory branches that become infected and diffuse and replicate. They don't go down, they go up. So then the next question is, well, in order to get a scare quotes significant alveolar infection, how many seeds do you actually need down there? And it will turn out that what matters the most of a seed of infection, which is any kind of deposition of an aerosol or some other, I'll talk about another possibility. What matters is that they're not so much, almost hardly at all, how big the seed is, how many infectious virions there are, but how many seeds there are. And so what we're going to calculate is, and I said this earlier, but the reason that the size of the infectious seed does not matter is because the replication is going to dominate the size in a short period of time. As I'll show you in a minute, we can calculate, given the infectability of the alveolar cells, the alveolar type 2 cells, how many seeds you need that are physically spatially separated in order to pick the percentage of alveolar type 2 cells would define a significant infection. Okay, then the result 4 there is that, and I'm going to talk about that in a little while, that you could either inhale aerosols with one infectious virion, or 5 or 10, or you could, it's well known that many people, in particular the people that are you'll see in the hospitals, they aspirate boluses from a nasal infection. But even so, what matters the most is the number of seeds that that are spatially separated. Okay, next slide. so as I said, this means that there has to be a deposition all the way into the alveolar space on the order of 1 million spatially separated seeds. If they're all together, it's not going anywhere. And if you think about the surface area of the alveolar space, it's 140 square meters. One little 1 mil high-titer bolus is not going to make an impact on 140 square meters. So you need many seeds on the order, and I'll show you in a minute how we quantify that. So we are strongly suspect that the way you're getting all these seeds is that you develop a high-titer nasal infection. That turns out to be sustained. It's very sustainable. And then you either aerosolize just like you exhale aerosols with infectious virions, so do you inhale them. Or these boluses, which we know people aspirate boluses into their lung, that those boluses, if they're say 1 to 1.5 mils, they will obstruct, they will not be able to take, when they go down into bifurcations, they're going to be too big to go down one of the 2 bifurcations by generation 3. So what we strongly believe is that those boluses divide, obviously not equally. Some goes one way, some goes the other branch. And then that happens on the order of 20 times, where a bolus will effectively aerosolize by the time it gets to the deep lung. There's a physics of why this bolus slides that I don't want to talk about now, but it turns out it's got super contrast physical properties, and there's almost no adhesion to the mucus lining the branches of the lung. So it raises a question. You need on the order of 1 million physically separated seeds into the deep lung. And the question that we have no answer for, but it's implied that why is there such a small percentage of individuals that get a high-titer nasal infection and subsequently aerosolize or aspirate that infection into the deep lung? That's a fundamental open question. Next slide, please. So here is the outcome of the modeling predictions for the alveolar type 2 cells. We're saying, okay, so many physically separated seeds arrive in the alveolar space. Turns out that alveolar type 2 cells are only maybe 5 to 20% of the surface area of the alveolus, and they're the infectable ones. And there's a very low probability of infection per encounter. So very different than the nasal tract. And then we just do the count in the lower right branch, the 2 left and the left and right, the first and second graphs down there are, let's suppose, pick the number, land it all at once in the alveolar space, or phase them in over a week. Imagining here that someone aspirates while they're sleeping at night for, say, 8 hours every night for a week. And then we calculate on the right, given those probabilities of infection and the percentage of infectable cells, what surface area of the alveolar type 2 cells is infected. And you can see that the number of seeds required is at least 10 to the 5th. And for a significant, whatever you say that is, it's on the order of 10 to the 6th to 7th. Next slide, please. So those are the sort of explicit results we wanted to show. Back one, yeah. What we're searching for, and I know we're all doing the same, is, and the question was asked to me by Juan Mesa, the DMS director of NSF, what is the tipping point? What are the conditions by which just below that threshold, you would clear the infection, versus just above it, you would transition to a deep lung infection that would require hospitalization. And what we're obviously, especially what Morgan showed and what others of you know, is that it's a combination of factors. It's not one effect. And we've been working on several that were raised by this work with folks that were connected by the NSF Rapid community that all got this 1 year rapid funding. One group at ASU, Lawrence Berkeley National Lab in New Mexico, and another group at San Diego State and Northwestern, in my group. And we're, again, just like Morgan was saying, we've got so many conversations going on through this community and others that we just keep sharing information. What Morgan presented is what we have not put in yet. We used to study how the immune system is cooperating, literally exploiting interactions with mucus to protect you against viral pathogens. But so far, what we've done is suppress that because of the novel coronavirus. And what many of the things that Morgan and others of you have talked about with immune responses, we now have to build and integrate on top of what I've shown you so far. Next slide, final slide, believe it or not. So there are open questions, right? That even prior to worrying about the details that Morgan has presented. And an open question right now, and this is a bit of a sensitive issue, is that we're now talking about advocating to send children back to school. Well, we don't really know why children are less susceptible. We don't know if they're actually getting an infection, but it's an asymptomatic infection. The data is mixed according to my Lung Institute colleagues on susceptibility of children. There's a strong suspicion that their immune system adapts way more rapidly than an adult. You all know this better than me. But anyway, what concerns me is that we don't have rigorous scientific basis for making a decision that's apparently about to be made nationally. Part of what we do as mathematicians and scientists is that we're putting bounds on every parameter I showed you in my model prior to an immune response, which we have yet to integrate in. And then we look at things like, what are the most sensitive parameters? Morgan mentioned something about that as well in her presentation. What lower or upper bounds of any of these parameter spaces define certain subpopulations that would make them more vulnerable? And those are the kinds of questions that we're mapping out even prior to considering immune responses. Anyway, I think a fundamental question that the two groups I talked about on the previous slide are really addressing is, using physics, chemistry, and physiology, why do some people propagate a high-titer nasal infection to the deep lung and others don't? Why would you aerosolize and down to the deep lung, whereas others don't? Why would you aspirate and effectively aerosolize aspirated boluses? There's a lot more conjectures for that scenario than others. Anyway, that's all. I hope I didn't go too far, but anyone who wants to connect with me, and I can, if I don't know what you're interested in knowing, I can surely connect you with the people that we know. Okay, thank you.
Terrific, thank you. Thank you, Greg. Any questions for Greg?
There are a couple hands up that I can see in the Zoom. Which you're, Jake.
Okay, let me look now. Yeah, I hid the math. I didn't think in a 10, 15, 20-minute presentation you'd like to be dragged through a bunch of mathematics. So trust me, we can hand you all the formulas and equations you want and show you the codes if you'd like, once we publish them at least. A bolus is, that's a question there from Ruchira. What that means is that people, if you have independent, many people literally aspirate nasal fluid into their lung while they're sleeping. Obese people are very prone to this. And so if it's the nasal fluid that's got 10 to the 6 infectious virions per milliliter, then those, 1 to 1.5 milliliter boluses, we call, that's what Rick Boucher's name for them is, they turn out to have very different physical properties than the mucus lining your respiratory tract. So the belief is that they slide. There's no adherence whatsoever. They have huge viscosity and viscoelasticity contrasts. And what we say is that's in a Cell paper from last year with Ralph Barrick and Rick Boucher and about 25 other people. What we're telling them is that that bolus must divide many, many times in order to have an impact on a deep lung infection. So anyway, I'm going down the list.
So sort of as a follow-up to that, is there data on any particular association with sleep apnea? And if so, what is the effect of using a CPAP machine on these?
Yeah, I can't answer that question about the CPAP machine, but clearly many folks that you will see hospitalized with COVID have apnea, and they are aspirating their nasal infection or have aspirated their nasal infection. But that's, I don't want to suggest that I'm deep in the weeds of the medical aspects of this. I just lean on my colleagues to give me as much information as they can possibly give me. But yes, clearly it's highly correlated with sleep apnea. Those folks are if they have a nasal infection, they're quite vulnerable. Let's see.
I think Fred.
Yeah, so John Rice is asking a question about the whole point of this is, what do we do to inform the medical clinical community at, you name the stage of this post-exposure? And Ronit Freeman is one of the folks that I work with, and Rick Boucher, who are seriously probing the question of, and my colleagues at Northwestern and San Diego State, why would one, what are the physical properties of the nasal fluid that would lead you to aerosolize those fluids, right? With even 5 infectious virions. And then if you knew that, and you could test their physical properties, what surfactants or what chemical, simple chemical treatment, what mucolytic treatment would you prescribe and show would diminish the aerosolization of that fluid? So that's a fundamental question. There's a lot of work done with macaques by Schuch in Germany that shows that not only do you aerosolize and aspirate to the deep lung, but you also do the reverse. The small bronchioles have way less elastic integrity, and they will close and open during breathing. And when they close and then open, they will aerosolize. And if there's an infection down there, that aerosol in the exhaled air will travel all the way to the upper respiratory system. So there's this amazing active transport from the nasal passages all the way to the deep lung and back. that's occurring and potentially occurring all the time. So that's just something interesting. I'm trying to look at more. Yeah, whether a humidifier is good or bad, I haven't studied the physics yet. I don't know what the properties are that would promote inhalation and exhalation. So I don't know how to modify them because I don't know what properties cause it. Now, there are many people in this community where there's an aerosol subgroup, and we've connected. And what I told them was, I'll tell you today why I really care about aerosols. And so presumably we'll follow up. But I'm more from the side of understanding the physics and the chemistry that would explain why you would have a tendency to aerosolize during breathing, and then how we would suppress that with mucolytics, for example. Other questions?
Yeah, I think the mucolytics, that's a double-edged sword, right? Because at some level, if you decrease the viscosity with the mucolytic, you might actually increase the dispersion of whatever it is that you happen to have. So
the ability to aerosolize. I mean, so that's the issue, is that the physics and chemistry of it is, and material science of it is, at the Reynolds numbers, the semi-turbulence of the air when you exhale and inhale, how do you aerosolize? That's not a trivial problem, by the way. A lot of people would like to imagine that they understand the physics of that, and I would argue that I'm seriously question that. So knowing how you would modify the properties of nasal fluid such that you would suppress aerosolization, I think it's a very interesting problem, right? And it's not what you would say is a solved problem.
So I'm afraid we're just about out of time.
Yeah.
Thank you very much, Greg. Those both yours and Morgan's talks were just fascinating. And I want to just mention two things. We had started discussing some possible competition, hackathon type events last time, a dream challenge like thing. Hopefully we can get back to that at a later meeting. And I should also mention that as part of the steering committee, we've started discussing, what a concerted actual research effort might be coming out of this group and how one might fund it. So that too, hopefully, will be discussed in all the subgroups. So thanks very much for attending, everybody. Thanks, Morgan and Greg, for these very interesting talks. And I will see all of you next week.
Okay, thank you.
Bye-bye.